So today I'm going to talk about a paper that was published in my lab in 2017. This is actually work I did myself, looking at the effect of insulin-like growth factor, or IGF-1, and how its function signaling through the receptor it binds to, and how that impacts ATR kinase check-1 signaling and UV-irradiated human keratinocytes. So just some basic background information. IGF-1 is an important growth factor, particularly in keratinocytes and in human skin. What we know from a variety of literature from the labs of Jeff Travers and Dan Spandau, these investigators have shown that as people age, the IGF-1 level, the expression of IGF-1 in the skin decreases. And in the skin, IGF-1 is produced in dermal fibroblasts, in the epidermis. However, the keratinocytes that are located in the epidermis are unable to produce IGF-1, but they do have IGF-1 receptors. And so the thought is that IGF-1 in the skin regulates how keratinocytes can grow and proliferate, and then potentially respond to things like DNA damage induced by UV radiation. And again, as I mentioned, as people age, it's been shown that IGF-1 levels decrease, and therefore IGF-1 signaling in keratinocytes is also decreased as people age. And that might account for some of the differences in terms of how these keratinocytes respond to UV, potentially could lead to increased mutagenesis, potentially lead to increased skin cancer. So this altered IGF-1 signaling may, at least in part, explain why older people are prone to skin cancer. And so work from a variety of groups in the past actually have shown that geriatric skin less efficiently removes UV photoproducts from DNA in the human skin. And work that had been published in the previous year, Dan Spandau's group had shown that if you manipulate IGF-1 signaling in keratinocytes in vitro or in human skin explants ex vivo, and in mouse xenografts in vivo, that this alters the rate of removal of UV photoproducts. So it looks like there's a defect in the rate of removal of UV photoproducts when IGF-1 signaling is not active in keratinocytes and in human skin. And so with that information in mind, I was curious about other aspects of the DNA damage response. When I was a postdoc in Aziz Sanjar's lab, I'd worked quite a bit on this kinase known as ATR. We know it regulates multiple aspects of the DNA damage response, particularly in replicating cells, where we know quite a bit of what it does. Regulating things like replication fork stability, replication origin firing, and other things that stabilize the genome during S phase. And so in this paper, what I want to do basically is to look at how ATR signaling is affected by the loss of IGF-1 signaling. And so in the first figure, in figure 1, what I did was to take telomerase immortalized keratinocytes. That's sort of one of the main cell lines we use in the lab. These are neonatal foreskin keratinocytes that were then immortalized with telomerase. Grown in culture, we grow them in defined medium, so we can actually manipulate some of the growth factors. And the first experiment what I did was to treat cells with your vehicle, DMSO, or a compound known as AG538. This is an IGF-1 receptor inhibitor. So it's similar to the effect so it can mimic the effect of potential geriatric skin by inhibiting IGF-1 signaling. And so what we did was, again, pre-treat with vehicle or DMSO for 30 minutes or an hour, expose cells to UV, and then look at the signaling phosphorylation of substrate proteins that ATR is known to phosphorylate. And so one of the main substrates most widely known is CHEK1. It becomes phosphorylated on serine 345 after DNA damage and after UV radiation. And so if you look at the vehicle treated cells, again, within 15 minutes or so, you start to see phosphorylation, peaks half an hour to an hour after UV, and then starts to decrease. In cells treated with AG538, we still see phosphorylation, but it is significantly reduced. Again, this is only a brief treatment of the compound with the drug prior to UV exposure. We also looked at CAP1. This is a heterochromatin protein. ATM is known to phosphorylate it. However, ATR has also been shown to phosphorylate it as well. And there's also interplay between ATR and ATM. So essentially both kinases could affect that. So, and we very similarly see a decreased level of CAP1 phosphorylation when the IGF-1 receptor is inhibited. In the graph there, we quantify that. So in the graph here we quantify, we can see that there's reduced levels of CHEK1 phosphorylation quite significantly. And this is after a pretty high dose of UVB. In figure 1A, this is 500 joules per square meter, a pretty lethal dose or pretty toxic dose. We also did dose responses where we used 0, 60, and then up to 500 joules per square meter UVB. And regardless of the dose, we see reduced phosphorylation. So for example, if you look at even a low dose like 60 joules per square meter, there's a lot more phosphorylation over here than there is in these cells. And so again, just consistent with the data in figure 1A. And the graph there just quantifies that. So one of the functions of ATR in CHEK1 signaling is to suppress replication, origin, firing. That's where DNA initiates along chromosomes. So to measure DNA synthesis in these cells, I performed an immunodoppler assay where I treated cells as before, exposed them to UV, and then pulsed the cells with BRDU, bromodeoxyuridine. This is a nucleotide analog, gets incorporated into DNA during replication. We can then purify genomic DNA from these cells, dot it on the membrane, and use an antibody against BRDU to detect the level of incorporation. We can also reprobe or probe the blots for total DNA on the blots. And they're denatured in when we immobilize them, that's why we use an anti-single-stranded DNA antibody. And so what you can see is with increasing doses of UV, we see reduced levels of DNA synthesis, so there's reduced BRDU incorporation into the genomic DNA. And consistent with the idea that we want to suppress DNA synthesis after UV exposure to potentially limit the likelihood of mutagenic DNA replication. However, in cells treated with AG538, although we still see a decrease, the decrease is not as robust. So in the graph below, what I've done is to quantify the level of DNA synthesis in the UV irradiated cell in comparison to the non-radiated cell over here in both cases. And so you can see, again, very nicely that there's a difference in the level of DNA synthesis. There's more DNA synthesis taking place when the IGF-1 receptor has been inhibited than in the normal vehicle-treated cells. In figure 1D, I've done a time course. Again, this response is actually a transient response. So if you look in the DMSO-treated cells here, again, cells are going to repulse BRDU at different time points. 0 is before UV exposure. And then immediately after UV, and you start to see a very rapid decrease in DNA synthesis. And the maximal decrease occurs, just a few hours after UV exposure. And then the DNA synthesis starts to recover. So by 6 or 7 hours later, we start to see DNA synthesis recover in these cells. However, when cells are treated with AG538, although again there is still this decrease, there is still this inhibition of DNA synthesis. It's not quite as robust. So again, there's more DNA synthesis taking place with inactive IGF-1 receptors than in vehicle-treated cells. So again, this was in telomerase immortalized keratinocytes. We wanted to confirm this in primary cells. So we took a grown this is another inhibitor. Sorry, I think I'm getting ahead of myself. That's the next figure. So this is cells treated with AG538 or with another inhibitor, this abbreviated PPP. For another compound, I always forget how to say the complete name of that compound. It's in the paper. It also inhibits that IGF-1 receptor through a different mechanism or through a little bit different means than AG538. Again, we see a partial reduction, not complete. Again, there's still phosphorylation taking place. But again, there's a reduction in the level of CHEK1 phosphorylation after UV exposure. And if we look at BRDU incorporation, again, there's more DNA synthesis taking place with these inhibitors present than without. And so, again, it looks like you need proper IGF-1 signaling somehow to properly suppress DNA synthesis after UV exposure. And so this is, next wanted to do was to look at this in the context of depriving cells of IGF-1. So again, these end-term keratinocytes, what we do to grow them is to we use a defined epilife medium, which contains a human keratinocyte growth factors, one of which is IGF-1. And so we can actually remove that from the kit, from the reagents, and grow cells without IGF-1. So what we've done here is cells were grown without IGF-1 or with IGF-1 and then exposed to UV. And so and then we can look at CHEK1 phosphorylation here. Again, in both cases, CHEK1 gets phosphorylated, but we see a more robust response. So it looks like it's maintained at a higher level later time points after UV exposure when IGF-1 is present. It's not quite as robust as the IGF-1 receptor inhibitor, And we're not exactly sure why. Part of it could be the efficiency by which an inhibitor can actually inhibit the kinase signaling, whereas depriving cells of IGF-1 or stimulating it might not be quite as robust. It's also possible that there could be off-target effects with the small molecule inhibitors, that maybe this is a true response here in this figure, but the inhibitors are a little bit more due to other effects of the drug. Nonetheless, we also looked at BRD incorporation by DNA synthesis, and you can see, again, the more IGF-1 you have in the cells, this is somewhat counterintuitive because we know IGF-1 is a growth factor, but the more IGF-1 is present in the medium, the more efficient the suppression of DNA synthesis after UV exposure. We've also done, this is at a single time point in figure 3C. This is with a time course. So again, we see the same kinetics, things get inhibited, then recover. However, again, cells without IGF-1, there's a little bit less. Again, the suppression is not quite as great at earlier time points. So again, in this figure, again, figure 3, we're looking in manipulating IGF-1 signaling by depriving cells of IGF-1 or not. So this is all, so far has been in keratinocytes, again, in vitro. We next wanted to move to a human skin system. So what we've done is taken abdominoplasty skin section. So this is skin discarded during normal surgery. These human skin explants, we can use them ex vivo. And this is actually, I believe, the first report to actually show that CHEK1 can become phosphorylated in human skin epidermis. And so what we did is to take human skin, expose it to increasing doses of UV. We typically have to use higher doses of UVB in human skin than we do in vitro because human skin epidermis is multiple layers, and therefore potentially, and this has not been looked at yet, but CHEK1 expression, for example, in the epidermis might not be equivalent in the different layers of the epidermis. And we hypothesize actually that CHEK1 may be more expressed in the basal keratinocytes at the bottom level of the epidermis, because those are the cells that have the potential to grow and divide and replicate, whereas the cells in the upper layers of the epidermis are differentiated and therefore may not potentially express CHEK1. But again, all this is hypothetical. We haven't actually looked at that yet. CHEK1 is known to be regulating the cell cycle. Again, CHEK1 stands for checkpoint kinase, and we typically think of checkpoints and cell cycle checkpoints in the context of cells that are actually moving through the cell cycle. So in the context of skin, that would be found in the lower level of the epidermis. Nonetheless, we can expose cells to increasing doses of UV and indeed see CHEK1 phosphorylation on serine 345, an ATR target site, and serine 296. This is an autophosphorylation site. So once CHEK1 becomes active by ATR phosphorylation, it can then phosphorylate itself. And so we can see both of those phosphorylation events in the human skin epidermis ex vivo. We've done a few time courses, and this just shows us in figure 4B here. Similar to what we see in skin or in keratinocytes in vitro, it peaks an hour to an hour after UV and then starts to decrease. So by 4 hours, the level of CHEK1 phosphorylation is decreased. So this is a transient response. So we next wanted to look at the effect of manipulating IGF-1 signaling on this response. And so what we did is to topically treat the skin either with vehicle DMSO or with 20 micromolar of AG538, this IGF-1 receptor inhibitor, expose the skin for 30 minutes or so, then expose cells to a couple different doses of UVB in relatively high doses, and then look for CHEK1 phosphorylation and CAP1 phosphorylation by Western blotting. And they're never quite as clean in skin ex vivo than in cultured cells. Nonetheless, you can see phosphorylation of these proteins in a dose-dependent manner in the vehicle-treated cells. In the AG538 treated cells, we see both a reduction in the amount of CHEK1 phosphorylation, so that's reduced here, as well as CAP1 phosphorylation, another substrate of the ATR and ATM phosphorylation. In figure 4D, we've quantified that. So again, we can see a reduced phosphorylation, particularly at the higher dose of UVB that was, again, reproducible in multiple independent skin samples. So we're curious, why this is different. So we know that one of the signals for activating and recruiting ATR to sites of DNA damage and replication stress is RPA. RPA is a single-stranded DNA binding protein. And we can actually look for that enrichment in cells by fractionating cells to enrich in associated proteins. So and what we've done in figure 5 is to go back to the keratinocytes in vitro, the enteric keratinocytes, treat with DMSO, the vehicle or ATF538, exposed to UVB, I believe it's 125 joules per square meter. And then at different time points, harvest the cells and isolate the chromatin and then do Western blotting for proteins that are bound to chromatin. And so what we see after different time points after UV exposure is we do see RPA becomes enriched on the chromatin, so it increases it particularly 1 to 2 hours after UV exposure. However, when we treat cells with AG538, although there is some increase, it's somewhat less robust. So there's somewhat less of an increase in RPA binding to chromatin. And since RPA association with DNA is a signal for recruiting ATR to sites of DNA damage, in this paper we hypothesize that this might be why the signaling is reduced when IGF-1 signaling is decreased. It's somehow altering an early step in the DNA damage response. We don't really fully understand how IGF-1 signaling impacts that, and that's one of the aims of my laboratory is to further figure this out. So with this data together, we put a model together. Again, this is based on both data here as well as other literature from other publications. Is that in younger individuals where there's sufficient levels of IGF-1, again, a young adult potentially, they may get exposed to UV that will generate DNA damage. However, they have a normal nucleotide excision repair capacity, or maybe a solid, good level of DNA repair activity. And that's can be shown by other groups. And we know that IGF-1, normal levels of IGF-1 seem to promote some level of DNA repair. Now, in this paper, we've also suggest that in addition to repair being affected by IGF-1 that and by UV, that ATR signaling is also affected. So again, as our data have shown in the paper, when IGF-1 is present or when the receptor is active, we get good levels of ATR phosphorylation check 1. We see a suppression, again, inhibition of DNA synthesis. And we hypothesize that this means they're less likely to be mutagenesis. Again, this combination of repair and suppression of DNA synthesis. It may also, if there's high levels of DNA damage, promote cells to undergo senescence. And together, this will limit the likelihood of cancers developing. However, when IGF-1 is deficient, so if we add AG538, for example, or deprive cells of IGF-1, when there's damage by UV, we know that there's decreased rate of nucleotide excision repair based on other literature, including by Dan Spandau. And this paper shows that there's somewhat less ATR check 1 signaling, again, a higher level of DNA synthesis than we would expect. So together, we think this inefficient repair and failure to suppress DNA synthesis may increase the likelihood of mutagenesis, of mutations occurring in these cells. And along with other data suggesting senescence is affected, this increased the risk of carcinogenesis in the skin. And therefore, again, we hypothesize that part of what reason why older individuals are prone to skin cancer is because of this deficient IGF-1 signaling that alters the DNA damage response, not only through nucleotide excision repair, not only through removing UV photoproducts, but also through the efficiency by which they can suppress DNA synthesis in epidermal skin keratinocytes.